All right, well, hi, I'm Sunetra Gupta from Oxford. Very, very pleased to be invited to this, what's it called? Meta meeting. And I was here for the last meeting in October, well, maybe that wasn't just the last one, but and learned so much. Essentially, I work on infectious diseases, so maybe I'm a little bit peripheral to the program, but so for me it's fantastic to learn about several of the different systems that are being discussed here. And what I'd like to do today is to give a little bit of an overview of some of the work that we've been doing looking at the structure of metabolic genes in bacteria, in the hope that perhaps this might trigger some new ideas and thoughts about how you approach some of your own systems. So, there we go. Right, okay, so my colleague Oliver Pybus and I co-teach a course on the evolutionary dynamics of infectious disease, and we sort of somewhat jokingly divide up the genome between us into, Oliver talks about how you can apply phylogenetic and phylodynamic methods to regions which are essentially under weak or no selection, and my bit of the course is more about bits that are under selection, which of course essentially comprises metabolic genes, structural genes, so on and so forth. But what, again, I urge the students to do is to consider these, to collect these into a kind of scheme where you think of transmission factors, think of the pathogen genome as containing transmission factors, virulence factors, and antigenic determinants as a kind of way of structuring your ideas about what these genes are actually doing, many of which, of course, there's quite a bit of overlap, indeed, in these functions. The same gene can contribute to transmission and virulence and indeed be an antigenic determinant. But overall, this kind of a framework is what my group has been using to try and understand the evolution of diversity at all of these levels in a very large range of pathogens. Today, of course, I will focus only on the bacterial pathogens. So essentially, of course, diversity can exist at any of these loci, in any of these factors, and the sort of traditional epidemiological models that we, most of you, I presume, will be familiar with, essentially assume that there is a total uniformity so there, in these factors, so there is no difference in transmission or virulence or among the antigenic determinants of the constituent strains or whatever it is that the members, the individuals within a population, pathogen population. And then this then leads to the standard SIR formulation, which I will not go into, which once you stick some births and deaths into it, provides you with the sort of classical kind of epidemic dynamics that does such a good job of, with some external forcing, must be said, in explaining the dynamics of certain pathogens such as measles, which indeed conform very nicely to this scheme, in that you can think measles certainly does have antigenic determinants that are conserved across all the different members of a population. In other words, what we target within when we get a measles infection is a part of the virus that it cannot really afford to change. It's an invariant part of the virus, and that lends itself to this treatment within a sort of classic SIR framework and allows us to understand its epidemiology through the employment of that kind of a framework. One of the other critical sort of concepts that comes out of even a very simple SIR type model is this notion of a basic reproduction number, again, that I'm sure many of you will be familiar with, which is a measure of the fundamental potential of a particular pathogen to spread within a population. I call it fundamental population potential, and the reason it's called basic reproductive number is it's almost, what it is an articulation of its maximum success rate. And as soon as people start becoming immune, this of course diminishes. But anyway, so it's a fundamental measure of the potential, transmission potential of a pathogen within a given setting. So you can start introducing variability into this scheme at any of these factors. Of course, you can start to introduce variability in virulence and alter the equations to incorporate variation in virulence by saying that the death rate out of those who are infected by one strain or the other is different, and this will immediately impact upon the basic reproduction number of the particular strains. So if you've got strain 1, which kills at a rate alpha 1, that's what now factors into the equation for its fitness, if you like. And once you start playing with models like this, where you now have introduced a little bit of diversity, you generally get a very clear outcome of these two strains competing in an environment, which is that the strain with the higher R0 always wins. Similarly, if you start to introduce variation in some of the transmission factors, once again, you alter the strain's R0, and in any such system, once again, you get the outcome that the strain with a higher R0 wins. This is classic competitive exclusion, and the reason it's occurring is because they are sharing the same resource. By virtue of having a common antigenic determinant, they're essentially trying to exploit the same resource. As soon as one strain infects an individual, that resource is taken away from the second strain, and therefore they are in direct scramble competition for these resources, and the strain with the higher fitness, if you like, the higher R0, always wins. So pathogen populations that contain antigenic diversity have had to get around this problem, and essentially the very reason that they can be antigenically diverse is because they don't have the conserved bits, the conserved determinants, the things to which we mount immune responses, if they are conserved, are either very weak in terms of the immunity they induce or don't exist at all. In other words, if you rank what we recognize, so any pathogen will contain determinants that are conserved and determinants that are variable in terms of what we recognize. But the strength of immunity varies according to different pathogens. So as I said, in measles, the bits that we recognize are highly conserved. They're located in its receptor binding region, and it just really can't afford to change them. But there are a number of other pathogens, ones that I mainly work on, which have the capacity to vary the regions that we principally recognize, the dominant antigenic determinants. What that means is that those systems can no longer be represented by a conventional sort of SIR model with differences in strain fitness, but because what we really are looking at is a situation now where the antigenic determinant itself is variable. So, of course, one can take the SIR model and proliferate the compartments and link them up in appropriate ways to also look at the interactions between different strains, now defined in terms of whatever they possess at, let's say, a particular antigenic locus. And if they happen to possess completely different elements, variants, which have no cross-reactivity with each other, these strains would circulate as independently transmitted strains. So you might as well be talking about mumps and measles, for example, if they are completely distinct from each other and don't interact. However, one can introduce, there may be some level of cross-immunity between two strains of the same pathogen, either because the antibody responses or whatever it is, whatever type of T-cell response, whatever it is that's recognizing one strain, may have some action against that's different variant at that same locus, or because, of course, there are immune responses to other parts of the pathogen that may be common, although weak. And this can be captured within an SIR framework by modifying the frequency with which an individual who's been exposed to a particular strain might become infected by a second strain. So you can just modify that risk of infection by a term 1 minus C, where C represents the cross-immunity between these strains. When C is 0, there's no cross-immunity, and when C is 1, cross-immunity is complete. And what you see in a simple sort of framework like that, when you allow cross-immunity to vary, is that when you have, again, absolutely no cross-immunity at all, you get a region of complete coexistence of the strains. The axes here represent the R0s of strain 1 and of strain 2. So you get complete coexistence when there's no cross-immunity. When there is total cross-immunity, you get competitive exclusion, such that the only strains that can coexist are ones that have identical R0s. And then in between, as you decrease cross-immunity, you kind of open up the region across which strains of different R0s can exist. So if they differ too much in R0, one will outcompete the other, but there's a sort of certain tolerance develops in the system of how much of a difference in R0 it can take. So that kind of a framework can be used quite successfully, just that very, very simple kind of idea, strains competing, a little bit of cross-immunity, maybe not, to understand the maintenance of antigenic diversity in a number of pathogen systems, and one of these is the pneumococcus. So Streptococcus pneumoniae is a gram-positive bacterium that, as many of you know, is carried in the nasopharynx, but causes, on occasion, quite rarely, invasive disease, including obviously pneumonia, but also meningitis and in rare cases sepsis, and this occurs when the bugs move across the barrier, they invade through the epithelial barrier, and this then results, as I said, in either pneumonia or meningitis. And this is a big problem, as I'm sure many of you know. It is one of the largest killers both of children and of adults. So this is right up there in what causes human deaths. So the pneumococcus is, as I said, a gram-positive bacterium, and if we're thinking in terms of what is its principal antigenic determinant, it would be safe to say that that antigen, that locus, would represent the capsule that it contains. So the pneumonia are surrounded by this thick capsule, and this capsule is highly antigenic, and it's the basis of a classification system known as serotyping. So different serotypes of pneumococcus, which are effectively different antigenic types, co-circulate in the population, and these are all stratified on the basis of what kind of capsule the bacterium carries. So this is just a sort of plot of the age structure of disease, just showing that it kills a lot of children and also a lot of adults. But what I want to draw your attention to is the fact that there are 90 known capsular serotypes of the pneumococcus. However, of these, only around 15 cause invasive disease. So if you look in a population and look at the proportion of invasive pneumococcal disease that can be attributed to these different capsular serotypes, you will notice a very strong overrepresentation of a certain set of capsular serotypes. So certain antigenic types are specifically associated with invasive disease, 14, 6E, serotype 1 in certain cultures. It differs a little bit from place to place, but there are some serotypes which cause disease. And this is partly because the capsule itself is a virulence determinant. This has been shown by through experimentation. But there are also other extracapsular virulence determinants which seem to specifically attach to these particular serotypes. So obviously this is a big problem. So what's, in terms of a vaccine, what the solution that we've come up with is to have a vaccine which targets these most virulent serotypes. And one of these vaccines, PCV7, which contains 7 of these serotypes, has been in use from about 2000 in the US and the UK, and also several other countries like Israel around the world, and also in sub-Saharan Africa. So here's a vaccine, PCV7, which contains a set of these serotypes, and the idea was, well, at least we're not going to knock out all 91 of these every day the catalogue grows, but let's at least target those 7 that are actually causing disease. Since the widespread use of this PCV7 vaccine, we've observed a growth in the frequency of non-vaccine serotypes, which, so for example, 19A has increased in frequency, which of course one has to, it can be placed within the context of the very simple model that I presented, by assuming there is some degree of cross-protection between the serotypes. Because if there were no cross-immunity between them, it would be hard to see why any change would occur at all once you use this vaccine. The fact that some change has occurred in the frequency of the non-vaccine serotypes suggests that there was some level of mutual suppression occurring. So with the pneumococcus, we're not here really. We are somewhere in this sort of region. There is some level of cross-protection. If there was too much cross-protection, then the whole system would collapse into a single strain. But what we have here is enough cross-protection so that you can get vaccine replacement, but not enough to destroy the diversity of the population. So that's kind of a nice way, perhaps, to think about, well, it's a framework, shall we say, to explain the persistence of different serotypes of the pneumococcus and why PCV7 would cause some of these to increase somewhat in frequency by, as it were, occupying the niches that were left, that have been vacated by the vaccine strains. So, in other words, we can define this system as a sort of single locus system and stratify it by alleles at a single sort of capsule locus. This is not, that's fine for pneumococcus, but this is definitely not how we can view a very large number of other bacterial pathogens. For example, its cousin, not very close cousin, the meningococcus. The meningococcus is a gram-negative bacterium, which is also frequently carried in the nasopharynx, and it causes obviously meningitis and also, again, septicemia. It's nowhere near as huge a problem globally as the pneumococcus, but it does cause outbreaks, as you know, and the case fatality in these outbreaks is very high. So it's still a major public health problem. There's just some pictures. So if you contrast the pneumococcus with the meningococcus, you have a completely different picture in terms of what are the dominant targets of immunity. So in the pneumococcus, as I said, the capsule is a dominant target and the serotypes are defined on the basis of the capsule. In the case of the meningococcus, it too has a capsule and it can be stratified according to that capsule into serogroups, A, B, C, much smaller number of serogroups, it's about 5 or 6. However, this capsule, whether or not it is immunogenic, depends on the type of capsule. So capsule serotype, the capsule A is quite immunogenic. So serogroup A does in some ways behave a bit like the pneumococcus, and you do get serogroup A occurs mainly in Africa, and you get this sort of epidemic pattern. In some ways, it behaves a bit like measles. But serogroups B and C, which are the ones that are circulating in the UK, for example, and also in North America, many other parts of the world, serogroup B in particular is not very immunogenic at all. It's thought because it resembles certain host proteins. Serogroup C is okay, and that's why we have a vaccine for it, but essentially the serogroups, the capsule is not the primary determinant of immunity against meningococcus. Instead, what we target when we're infected with the meningococcus are certain outer membrane proteins that stick out of the outer membrane and perform a number of different functions. So, so what we have here is a situation where, yes, we've got the capsule which determines the serogroup, and these are the names of the major serogroups that circulate. But actually, what we need to focus on, if we're trying to understand the epidemiology and the evolution of Neisseria meningitidis, are these outer membrane proteins, which I have drawn in theoretician's cartoon form here, which I will return to in a minute. You see why I did that. So, how can we visualize this? What's the appropriate kind of model for this kind of a system? Obviously, we can't just think of this as, what I should say, of course, is that, so we have a number of outer membrane proteins, and even with, which are highly diverse, and even within them, we have regions, different epitope regions that we recognize, which are in themselves very, very diverse. So we can't use a single locus system to understand the population biology of the meningococcus. What we need is a multi-locus model. And this, of course, is a cartoon of the very simplest multi-locus model we have, with two alleles at two loci. So what we did many years ago, about 20 years ago, was to develop a multi-locus model for pathogen diversity in which we characterized each possible strain by the epitopes or the alleles that it contained at different loci, and this is showing it obviously for the simplest case with the sort of two locus, two allele system. The scheme that we developed was not a simple SIR, just because you try and do this in SIR and you just end up with a whole mess of equations. So we developed a different scheme in which we allowed the compartments to overlap, and here the compartments Z represent the parts of the population, the proportions that are immune to a particular strain, let's say AX, so ZAX is the proportion that's immune to ZAX, and this equation just describes how you get to be immune, which is by being infected by ZAX, and then you die, because we're assuming that immunity to these to the strain is lifelong. In this system, you need to also figure out what proportion of the population is immune to any other strain that might be sharing alleles with ZAX. So in other words, people who are in ZBX or ZAY, because they share alleles with AX. And this is important because when you're calculating the part of the population that's infectious with strain I, the way you do that is by saying, well, anyone who's never seen anything that's remotely looks like AX is obviously fully susceptible. And then those individuals who have been exposed to something that looks like AX or shares any one of the alleles of AX, but not AX itself, have some form of immunity to AX. And just like in the very simple equations, the single locus model, you can modify that. You can include the strength of that immunity by modifying their infectiousness with the term 1 minus gamma, where gamma is the measure of the cross protection between strains that share alleles, and therefore is a sort of proxy for the intensity of immune selection operating at these loci. So this is just a way of summarizing that central assumption. In other words, what we're saying is that individuals who are immune to a particular strain obviously can no longer be infected by that strain itself, but these individuals are completely susceptible to a strain that doesn't share any of those alleles, because we're assuming that immunity is developing in a variant-specific manner to these particular variants. And then the central assumption is that strains that share alleles that have an overlap in their allelic repertoire with the thing to which an individual is immune will be blocked to a degree gamma. And that gamma is, as I said, the strength of immune selection. So if there's no immune selection, in other words, if this is pretty much close to, if gamma is close to 0, so that these things can infect this individual without much problem, then you just get the strains co-circulating, as you might expect. However, what's interesting is when you put this model under strong immune selection, so let's say you make gamma high, with a maximum of 1, what you get is a self-organization within the pathogen population such that you get the dominance of a subset of all possible strains, and the strains that dominate are ones that form together what you might call a discordant set. In other words, they don't share any alleles. So you get the dominance of a set of strains that don't share any variants at the relevant loci. Why is this? Well, of course, this is simply another form of competitive exclusion. These strains can coexist because they don't share alleles at any of the loci, whereas they will suppress, in fact, their own progeny because these strains share alleles with the dominant strains. So what you observe basically is the self-organization, the spontaneous self-organization under immune selection of the system into either one of these states, as represented here in this very simple cartoon, but you can extend this to as many loci, as many animals as you like, and you will get the same answer, that only if under strong immune selection, only one of the possible discordant subsets that you can create from this set of all possible combinations will dominate. And this will be the case even under full recombination and very high mutation. So you get
alternative stable states?
They can't, well, this is actually an extremely stable state, so it's very hard to invade this state, but they are alternative stable configurations of the population. But to get from here to here is, when there's strong immune selection, is very difficult. What we've also shown actually is that if you have weak immune selection, I'm not showing that today, not weak, if you have weak, you don't get any structure, under quite a broad range of parameter space for gamma, what you do get is a flip between this and that, and you can actually get very strong chaotic dynamics as well. So we use that particular result actually to explain influenza and HIV and pathogens like that, but I'm not going to go there today, but we can talk about it. But under strong immune selection, you will get this self-organization into non-overlapping combinations of antigenic variants. And so this is a paper, this is a result Martin Maiden and I published almost 20 years ago now. And essentially what we were saying is that if you look at a pathogen population with diversity, diverse antigenic determinants, you should find co-circulating strains that don't share any of these variants at any of these loci. And what we were able to show that this was the case with at least one meningococcal protein, which is porin A, which is, as I said, a major surface protein, and at the time was a novel vaccine component. And as I said, some of these proteins contain a hypervariable, and within the protein itself, you can identify different regions which are hypervariable. And that's certainly the case with porin A. And so going back to my silly cartoon, if this is porin A sticking out of the membrane, porin A has loops, and on two of its loops, in one loop you get this hypervariable region 1, another loop you get another epitope region, which is VR2, which is also hypervariable. And Martin Maiden, Ian Fever, and other colleagues at the time were able already in the early 90s to sequence these regions, which now like nothing, but in those days was really quite a big deal. So they had some information on the variation in these regions and how they associated in a population, in an isolate collection, which is at the time, which was quite small actually, it's only about 220 isolates, which were collected from meningococcal patients with meningococcal disease, and so they typed VR1 and VR2, and you could see that what they were. I mean, this is how we got together actually. Is I had the theory and Martin had the data. Martin Ian had been sitting there looking at this, saying, "Why does it look like that? Why don't you have more of a random association between VR1 and VR2?" And so we used the theory of this data to publish this paper quite a long time ago now, in which we were saying that porin VR combinations are non-random and also, crucially, non-overlapping, and this is because of immune selection acting to segregate them into these non-overlapping repertoires. So of course this was instantly shot down by everybody, but for various reasons, partly that these were disease isolates and also that they were close together, so they couldn't really rule out structural causes, etc., etc. So we spent the next 20 years or so gathering data on carriage isolates, longitudinal isolates, looking at other proteins, etc., etc. And now I think we have a large enough collection and have looked at a number of other proteins. For example, here, this is work done by Eleanor Watkins, whose work I'll come back to again in a minute. But this is just looking at those porin alleles against a different outer membrane protein, the FET-A allele, which is also immunogenic. And there too you can see this non-overlap in the associations between the variants. So we are feeling, 20 years down the line, fairly comfortable that that theory is at least consistent with what we observe in the meningococcus.
Okay.
So, yeah?
Can you explain a little bit how to read this figure?
Okay, so this is, the way to read this figure is to think two loci, lots of, a two locus system with lots of alleles at each locus. So what the theory predicts is that you will see a kind of diagonal signature of immune selection in this population, and that is, and we're saying yes, that is what you see. You see unique combinations arising, which are, obviously some of it is due to population neutral processes, but the fact that they're non-overlapping, and also if you look at the other genes, they don't show that kind of, obviously there's a lot more to the actual proof of, yes, this is immune selection, but this is, I'm just saying, take it from me for the moment and go and read the paper if you want, on, I'd like you for the moment just to have faith that this is, we think, signature of that kind of immune selection acting to segregate them into non-overlapping combinations. And this, I think I'll just skip over. This is also just to say that we've verified this using also stochastic methods, and one of the things those stochastic models also show is that you will actually, to achieve this kind of non-overlap, sometimes you will lose alleles. The population will actually lose some of its potential diversity. And this is work done by Caroline Buckey, which shows exactly that. And these are actually porin, that they are the numbers of loci that you see at porin A, VR1, VR2, those two variable regions, and that FET-A locus, and in different parts of the world. And you can see that although there's a huge potential for variation, they're always about the same, because that's what preserves that kind of non-overlap between the regions. Okay, so now finally on to metabolism, or indeed, well, before we get to metabolism, let's just let me just talk about, so I'm going to get to metabolism, but before I get to metabolism, I'm going to step back a bit and just talk about another scheme of typing the meningococcus. So where we've got to here is we've said that immune selection is causing pathogen populations to segregate into non-overlapping combinations of antigenic determinants. That's fine. Well, what about the rest of the genome anyway? So, as you know, the other very common way to type meningococcus, well, any bacterial pathogen species is, which is also developed by Martin Maiden and colleagues, is this multi-locus sequence typing technique, which, oops, sorry, this is supposed to show a picture of the housekeeping genes. So I'm sure many of you know this already, but essentially this is based on typing 7 or more, a certain number of housekeeping genes, and then using that to barcode each strain. And one of the things that you see when you, so this is an alternative level of structure that we decided we wanted to try and explore or understand. So one of the things you see in pathogenic bacterial populations anyway, when you do this MLST typing, which is of course originally developed as a sort of to understand the kind of clonal framework of these populations, and therefore one of the, in the language of MLST, one of the ways that these MLST types are described is as clonal complexes or lineages. And what you do see in most populations is the existence of certain central genotypes, which seem to persist over long periods of time. So let's call that the persistence of lineage problem. Now, obviously, the way people thought that happened before was because they assumed there wasn't much genetic exchange going on. But in fact, the very existence of some of the single locus variants of these central genotypes shows you that it's happening all the time. So how are these multi-locus, these lineages or central genotypes, common MLST types, maintained? Not only that, we also know that certainly for the meningococcus, and also it turns out from, again, most other bacterial pathogens, that only certain MLST types are specifically associated with invasive disease. So just as I said, the capsule serotype in pneumococcus, there's only certain serotypes cause invasive disease. With the meningococcus, certain MLST types seem to cause invasive disease. So this brings up, so here, this is finally the scheme of MLST. And the questions Martin and I thought we might try and answer were essentially these. How do lineages then emerge and persist? And why are some lineages associated with disease? And the answer that we came up with, and this is where we finally tie back into metabolism, is that these genes, which are of course involved in metabolism, I don't think anyone would deny that, actually determine the fitness of the bacterium. So the variance, the variation here is not neutral in these genes, that they have some role, that they're co-adapted to essentially define some form of metabolic type. And among these metabolic types, you have some which are more successful than others. Not hugely more successful necessarily, but successful enough so that when they're competing with each other on the background of the same antigenic variants, one of them will outcompete all the others. So what you have here is now, using that same scheme, you have obviously the antigenic types being characterized in a multilocus manner, but you also have the housekeeping genes or some sort of construct of metabolic type, which is now contributing to transmission. And we'll come to, and then if you throw into this mix, so essentially, yes, so what kind of interactions are occurring here then? Well, obviously we have the immunological interactions occurring at this level, but what we also assumed in this, in setting up models that included the metabolic type and the virulence factors, that these two also underwent some form of competition, which you might call direct interaction, direct ecological competition. And that's because they are sitting in the nasopharynx, obviously, thereby occupying prime space, but also eating certain sugars and attaching to different receptors. So one can make the assumption that different metabolic types, or strains that possess the same metabolic type or the same virulence factors, will also compete strongly with each other. So if you combine these into a model, what you get, and this is work by Caroline Bucky, which we published in 2008, what you get is, yes, you first get immune selection, sorting the system out into different non-overlapping clusters of antigenic genes, but then within each of these antigenic islands, if you like, you get competition between the metabolic types. Plus also, it's because of competition within the same metabolic type, it pays for this whole system, for an antigenic type to not share the same metabolic type as another antigenic type. So you have all these processes occurring, which then leads essentially to the population being organized into different antigenic types, each with their own metabolic type, and only some of which have sort of extra virulence factors attached to them by virtue of, and this depends very much on where the actual transmissibility that that metabolic type confers, because certain metabolic types, if they're a little bit more transmissible, can afford to have a virulence factor, which essentially will may contribute to transmission, but also may reduce transmission. And this is indeed, this is a terrible slide, but this is indeed, this, let me just show you, this is the letter. I don't know why these aren't quite well, but this is indeed what you see in the pneumococcus, as I said, as I showed you before, and also, going back to the streptococcus, what you see in the streptococcus. So here are the antigenic types, those capsular serotypes I talked about, and if you can do MLST on the streptococcus, on the pneumococcus, and you will also get different MLST types, and in various data sets you will see that particular serotypes always associate with one or possibly two metabolic types in a manner that is, again, non-overlapping. And what this bad slide is supposed to tell you is that this is something that you can observe across a whole range of different bacterial pathogens, where you can define the antigenic type according to whatever is the dominant target of immunity, and then you can define you can certainly look at the MLST type anyway, and you will always see this again, another level of non-overlap between antigenic type and metabolic type. Okay, so I'm blithely calling the MLST type a metabolic type.
So is the mapping really one-to-one? Like if you have a cluster in the east, this E-bars diagram, you have a cluster.
Yeah.
All of them have the same
Antigenic
type.
Yeah, yeah.
So it's
Roughly, that's what this is showing, essentially. Yeah, this is, yeah. So if you take strain 14, they will all be either, in this case, there's two STs. Yes. Yes, that is true. Not just in the pneumococcus, not just in the meningococcus. And they can switch, they can exchange, they can move around, so it's not as if they sort of started that way. There's some immutable connection between them.
It's not surprising because this Neisseria, for example, is so recombinant.
Yeah, well, that's exactly what this is. This is why you need this theory. It's not, they're not stuck together. They are being forced to.
Within the clusters. So my understanding was that a cluster is not a clonal expansion, but it's a group of.
That's why, this is why we need this theory. This is telling you why.
But these are clones then?
They're not clones.
No,
they're not clones. critical point. They're being thought, they exist as non-overlapping combinations of antigenic determinants and metabolic types due to immunological and ecological selection operating on the system, not because they're clones. No, this is not to do
No,
with the
but I mean, just from the
population.
sequence perspective, each ST is, well, if I understand it, you're saying these antigenic types are homogeneous within the ST. So if you just look at that locus, this is a clonal group.
That's not how I would define clone, but yeah, that if you just look at the capsular locus you will see the same thing in each. Yeah, can I just move and then we can, yeah. So, but essentially, yeah, borrowing somebody else's slides always leaves you with strange animations. So, but essentially, one of the things you might be questioning is why are we calling an ST a metabolic type? Now, there's no doubt that the housekeeping genes are involved in metabolism, but in what sense do they constitute a metabolic type? And to answer that question, Eleanor Watkins performed this extraordinary feat this summer, also to answer some referees who were looking at this paper, wouldn't believe us, for good reason perhaps. And what she did is she looked at some whole genomes of 616 pneumococcal genomes that collected in Massachusetts, and she basically identified every single locus that that could be involved with metabolism and transport, getting about 819 total, and analysed the allelic diversity of each of these loci. So what I'm going to show you now is a very colourful diagram in which each line represents a locus and each colour represents sort of allele number at that locus as per the genome comparator tool, using the genome comparator tool. And this is what she saw. So if you take the pneumococcal genomes and you stratify them according to their antigenic type, and then not just look at their MLST, but look at all those genes, you still see a huge concordance across each antigenic type. So in other words, within each antigenic type, the likelihood of having the same allele at that locus is extremely high, which is what this graph here shows. So this gives us confidence that, that there are actually such things as metabolic types, and that that actually the reason why MLST works as such a good scheme is because, of course, each MLST, each of those loci are actually embedded within this scheme. But what we're looking at here are these co-adapted metabolic and transport genes, which it's almost, they've undergone character displacement and then, and so have they have become these distinct metabolic types, which then, due to ecological competition and immune selection, are associating specifically with particular antigenic types. Oops, there we go. Okay, so one very last thing that I'd like to address is how, so we think that this model is a good baseline descriptor of what's happening in many pathogenic bacteria, in that you have a unique association between capsular serotype, or the antigenic type, and the metabolic type, as represented in the pneumococcus by the capsular serotype and the multilocal sequence type. And then you also have only certain of these particular strains being virulent. Now, what we've been using this framework to try and understand is what's actually happening as a result of vaccination in the pneumococcal population. I showed you a slide where we saw the growth of certain serotypes after, non-vaccine serotypes after vaccination. And that's fine, that can be easily understood in using very simple models. But another thing that's happened since vaccination is that some of these non-vaccine serotypes have started acquiring the MLST types of the vaccine serotypes. And this is a very good example, which is 19A, serotype 19A, which is increased since vaccination, its MLST composition has changed dramatically. So before it used to mainly have ST199 attached to it, and now it mainly has ST320, which I think comes from serotype 4, which was in the vaccine, and also ST695, which was associated with serotype 19F in the vaccine. So we can use our model to understand what's going on here. This is just a slightly different way of representing it. So if you have, this is a visualization where you have A, B, and C as the antigenic loci. Let's call it 1, 2, 3 as the metabolic loci. This is a very simplified one. And you also have the virulence. We have an extracapsular virulence locus here, which is either plus, which means virulent, or minus, which is avirulent. And what the model that I already showed you does is it, because of immunological and direct resource competition, this population collapses into, in this case, 3 types where you have non-overlap, as much non-overlap as is possible here. You've got A1, B2, C3, and only A1 is virulent, whereas B2 and C3 aren't. So you see that kind of population structure that I showed you in pneumococcus. Now what happens when you vaccinate against serotype A is, in this model, is you knock out that strain, you knock out serotype A, so you're knocking out that strain, but instead of just leaving these 2 strains in circulation, what happens is metabolic type 1 and the virulence factor shift across to serotype C. So why this is happening is basically because in this system, metabolic type 1 is fitter than metabolic type 3, and also C minus, 1 minus, so 1 minus is better than 3 minus, but then also 1 plus is going to be better than 1 minus because this strain already has the virulence allele, the minus virulence allele. So what you have here is a combination of competition and also this selection acting to create non-overlap between these between the circulating strains. And so if you start off with this combination, you vaccinate against serotype A, what you'll do is select for a different strain which has the same serotype as C, but now you've had the transfer of the metabolic type and the virulence type over to that strain. So that's what we think is possibly underlies this observation that serotype 19A has acquired the STs that belong to serotype 4 and 19F, and you can actually also see that in this whole genome analysis. You can see how 19A has now acquired, this is 19A before vaccination, and 19A post-vaccination still contains some of its original type, but also some of the metabolic type of 19F has shifted over to 19A. So you can see that in the whole genome. What about the virulence allele? We said that the virulence would also shift, and there is some evidence indeed of a particular pilin, a type 2 pilus, which has increased significantly in association with serotype 19A, again, since PCV7. So before vaccination, this pilus was seen, and indeed pilus type 1, mainly seen in association with the vaccine serotypes. Since vaccination, we've seen a massive increase in PCV7. So basically, I'd just like to get you to, I hope some of these ideas have made some sense to you, and I just think it'd be interesting perhaps to think about whether this conceptualization of, not necessarily a bacterium, should we say, as containing these different factors, and of thinking of competition as acting in different ways and in different, for different durations at these different levels, whether this can perhaps help us in a wider context to understand bacterial communities. So So I'd just like to thank once again Eleanor Watkins and Caroline Bucky, two extraordinarily talented graduate students. Well, Caroline's now professor at Harvard, but Eleanor's still a graduate student. Worked with me, Martin Maiden, of course, long-term collaborator, Bridget Penman, Ben Ashby, and Adrian Smith, been very useful in formulating these ideas. Thank you very much.
So yeah, I actually thought a bit more about it. And I understand the sequence types. These are so two sequence types could have just one nucleotide difference. Is this correct? Or in the 7 or 6 alleles?
Yes.
All right. So now I understand better. So it's really that you're genotyping with the sequence types quite well. You have 6 alleles sort of sequence, and then you're seeing this one-to-one mapping with the antigens.
Yes.
But
which we don't think is something that is arising due to processes, neutral population genetic processes. So the other camp will say that no, that's just neutral. That's just structure that's arising out of neutral processes.
But
and we're saying no.
Isn't it easier to assume that these antigen genes are recombining really fast? I mean, these are basically in genomic islands where they get the rates of gene gain and loss are enormous. So actually, there is a recent paper by Eugene Kunin. He says they're like 10,000 times as fast as the nucleotide substitutions. So then, if this is true, then this explains directly this result. But I don't understand why we need to assume anything about selection on the rest of the genome.
What do you mean? I thought that how can you have any structure if there's so much recombination?
Because of gene flow boundaries. But the recombination, the
but
boundaries...
there aren't any. I mean, we know that they can. So, for example, with 19, the fact that 19A is now...
It depends what you call structure, actually.
Well, I'm calling structure population structure. So what I'm saying is that 19A, that there's no barrier to 19A carrying serotype 695.
That depends what you call population structure. And for me, that's a
genotypic
clustering. So clusterings of STs. That's what I would call population structure.
Clustering of STs. Oh, why there are lineages.
Yeah, and that's
so how does that? So that's another issue, which is, I think, also addressed by thinking about them as metabolic types, which is that some are more successful than others. So that's another level. Is that what you're...
No,
I mean, genotypic clusters. So groups.
Can I have a question?
Yeah.
Okay.
So just to be clear, so I know nothing about pathogens, but presumably some of them live as commensals on your throat and some of them get to the age of a kid.
Yes.
So do we get coexisting commensals? So if I was to look at the back of our throats today,
Yes.
we'd find within us, within any one of us,
So what we're
cancers.
saying is that that coexistence is only possible among strains that don't share metabolic types. So that's why you get this clustering or the discrete metabolic types emerging and also associating specifically with particular antigenic types.
Okay, so the reason I was asking was if particular individuals only carry particular antigenic types, then there'd be no reason to have completely distinct metabolic strategies, because you'd be sorted by your metabolic, because two antigenic types would never meet. You'd have one or the other. But if they coexist, then you could say that then that would explain why each one would need to have a distinct metabolic type to which presumably coexists in the context of all the
They coexist. So metabolic antigenic types can coexist because they don't induce cross immunity.
Yeah.
So if you're infected by serotype A, you can be infected by serotype B.
Okay.
There is no barrier, so that's why they're coexisting. But you can't be infected. What we're assuming is that there's a barrier to being infected by serotype B if it has the same ST as serotype A. Okay. So serotype B then acquires a different ST. So that's why you get this connection between the antigenic type and the metabolic type.
So what we've got is a number of coexisting strategies in the genomes. And when you eliminate one of those strategies with a vaccine, then maybe you create a space for another strategy to arise,
That's
which is
exactly-
actually an amalgam of the two.
That's exactly correct.
Yeah.
Correct. That is exactly what we've shown, is that it's the removal of one,
Creates room for
it just reshuffles into another
yeah.
combination that works. That's exactly
So in this whole genome analysis,
Mm.
you could also look at all kinds of other genes, and you should find that some of them would be
Yes,
associating and some wouldn't, and that
that's
tells you which
right.
ones
So
are
let me see if
selected.
I've got,
Right.
do I have that picture here? That's what I was hoping I had. This is where, I don't know, for some reason. Yes,
Oh, okay.
there we go. So the metabolic genes, if you just look at the linkage to equilibrium, we also looked at things like mutual information, have, well, they do seem to have a different level of LD than the non-metabolic genes. It's obviously there is a clonal there is some population history here, so it's always difficult to completely reject the null hypothesis when it comes to these sorts of studies, I think. But I think you could, yes, I think obviously that's an obvious thing to do. So, yes, it's, I mean, what we're putting out here are some newish, new ideas in the sense that, can you really think of these things as having metabolic types? And the purpose of this exercise is very much just to put these ideas out and see if I think that's what we do best, is come up with hypotheses that then are hopefully testable. By no means are we saying that this is how it is, but hopefully we can drill down further into this, but with more data. As more data become available.